Welcome everyone, my name is Anne Lord, librarian at the Australian Wine Research Institute, and it's my pleasure to welcome you to today's webinar. It's also my pleasure to welcome today's presenter, Dr. Anna Carew from the University of Tasmania. Dr. Anna Carew is a research fellow in wine science with the Tasmanian Institute of Agriculture, based at the University of Tasmania. Anna joined the wine science effort in 2010 to undertake a PhD developing a novel process for Pinot Noir winemaking based on a microwave heating of grape must. Since 2014, Anna has been funded by Wine Australia to trial microwave maceration at commercial scale, and more recently to lead a project examining the key factors driving stylistic distinctiveness of Australian Pinot Noir wines. Prior to joining the wine community, Anna worked as a water microbiologist in sustainable consulting, as a university lecturer, and in engineering education. Before I hand over to Anna, I have a couple of quick points as to what to expect for those who are new to AWRI webinars. The format for today is a short presentation followed by a Q&A. The webinar is being recorded and will be available to view via the AWRI website. It is important to remember that webinars are interactive and that there are a number of ways to get involved. To ask a question, please type into the question section of your control panel in the bottom right corner of your screen. For those with access to a microphone, there is also the option of speaking directly with our presenter by clicking the Raise Hand button. This is located in the top left section of your control panel. Please feel free to send through questions at any time, as well as during the Q&A session, and I will get as many questions through to Anna as possible. Finally, if you would like to get involved through Twitter, please use the Twitter handle @the_AWRI. Okay, that's enough from me now, and I'm going to hand over to Anna to tell us more about today's topic.
Hello everybody, and thank you so much, Anne, for that terrific introduction. I'm hoping that we have a few people listening in today. I do understand it's been a fairly difficult 24 hours in South Australia, and my thoughts are with you all. I would like to really take a moment to thank the Australian Wine Research Institute for providing us with this opportunity to share some of the work that we've been doing at the Tasmanian Institute of Agriculture, and also I'd like to take the time to acknowledge the contributions that I'm going to be sharing with you today come partly out of my work, but all of that work is done in collaboration with colleagues. So colleagues at the Tasmanian Institute of Agriculture listed on the slide there, Dr. Bob Dambergs, who's with Wine TQ. We have a particularly strong collaboration with AWRI through Dr. Paul Smith and Dr. Karen Binden. Angela Sparrow's work will also feature briefly, and we have some work in here from Dr. Kai Knerzer and Mr. Piet Sviergon from the CSIRO at Werribee. So, innovative Pinot Noir maceration. Why are we interested in investigating novel maceration, particularly for Pinot Noir? Perhaps it's obvious. We are a cool climate state. Everybody suffers from challenging vintages, but what we're interested in particularly and motivated by the Tasmanian Institute of Agriculture is looking for options that winemakers can use at times when they are facing challenging vintages. We're certainly not dyed-in-the-wool techno lovers, so obviously we wouldn't recommend intervention unless it's needed, but good for winemakers to have those options there when they need them. So, challenging vintages. Years when the yield is very high, or years in which phenolics are poor. Obviously some of the winemakers that we work with also have blocks where the phenolics can be quite a challenge for Pinot Noir. In Tasmania, we talk about 4 seasons in one day. Weather variation is quite strong, and we face quite a lot of disease pressure during the time that we're trying to get fruit off. And so we may sometimes have to harvest fruit at a time when it's not perfect because we're facing a week or two of rain and we really need to get it in. We've also, all of us have recently seen, in recent years, seen quite strong impacts from what we see as climate change or climate change associated impacts. So we've been seeing an earlier harvest date for many, many years now, but we're really starting to see a crunch in terms of vintage compression, where many of the winemakers that we work with in Tasmania, but also in cool climate areas on the mainland, are reporting much contracted periods of time to get fruit through their facilities or wineries. So under those conditions, investigating novel maceration is really important to feed into production efficiency and maintaining quality under those kinds of pressures. We also are interested in novel maceration because the Australian wine market, of course, faces challenges. We need options to diversify wine style. Anything that we can do to allow winemakers greater control over the style they're producing, and possibly quality, although I guess we often talk about it more as a style choice rather than necessarily an objective quality, and more efficient production, given that we're facing challenges from other New World export markets that we want to inhabit. One of the reasons that we're very grateful in Tasmania that we're a reasonably small operation and that we work very much hand in glove with our industry partners is that we get clear messages from them about some of the problems or doubts that they may have about novel maceration processes. So on the bottom of the screen here you can see some processes have been associated with quite poor quality wine or fruit, and for example thermovinification in Europe has tended to have that reputation. Many processes involve high capital investment or maintenance challenges, and we certainly don't want to be recommending something that's going to involve a technician coming out from Germany every time it pops a bung. Some of these technologies are relatively unfamiliar in Australia, although we do have quite an international winemaker base, so that's perhaps less of a concern. And we're also very aware that anything we propose needs to offer a decent business case, and while things are still in development, obviously we wouldn't be recommending industry pick them up till we're fairly sure they're going to be robust and reliable. Okay, so a little bit of, this will take you back to uni if you enjoyed a stint as an undergraduate student, but this is a little bit of theory. So red wine maceration is really about mass transfer. So the objective here is to access the compounds that are trapped within the physical structure of the grape material and to release them into the liquid phase, into the juice. So on the bottom left there you can see a schematic of a dear little grape cell, a dark green cell wall around the outside. We know that phenolics are located in very many places within that cell. So we have phenolics in the cytoplasm that are floating free that we might want. We have some that are held inside the vacuole, floating around in the vacuole. We also know that some of the phenolics we might want to extract are actually associated with or attached to, loosely speaking, attached to some of the inner surfaces of those structures. And another consideration, which we don't know everything about this, but it's certainly something we're aware of, is that there are different ways in which phenolics are bound to some of those inner surfaces. So hydrophilic, hydrophobic and hydrogen bonding are all important factors to take into account when we're thinking about how to transfer the compounds of interest out of that structure and into the juice. Let's have a look at grapes. So here is a delightful little grape that we've sliced in half. You can see here the skin, and of course you can see all of the phenolics, or the colour phenolics anyway, that we're interested in just diffusing there into the pulp. When we get a little bit closer, if we take a tiny section of this skin and really look at it under the microscope, you can see here very clearly the vacuoles, and in this slide this is Pinot Noir, fresh Pinot Noir grape skin. You can see the vacuoles have colour compounds in them. This is where the anthocyanins would be trapped, and also within that structure we know there are different species of tannin that are trapped that we would really like to get out. This is a picture of that type of grape skin after 8 days worth of fermentation, and you can see that there is still some residual colour trapped in the structure, but the structure has changed quite remarkably. So that's what we're trying to achieve. We are trying to manage the process of mass transfer. Obviously the Tasmanian Institute of Agriculture has had a very active grape and wine research program for decades now. I joined in 2010, and since that time the work that I've been involved with has looked at some of these maceration processes. So our standard process is an on-skins, usually 8 days on-skins control. We've looked at cold soak, extended maceration, enzyme treatment. So these are some of the more routine things that you would be using in industry. We've also developed or pioneered a couple of quite novel processes, thermovinification using microwave, which I'll speak about today. There's a cap shredding process, which our colleague Dr. Angela Sparrow developed during her PhD. I won't speak about that today, but there's plenty on the internet about her ACE macerator, and it's very interesting to look at. We've also looked at must freezing, high SO2 dosing, what we call inline CO2 simulation, and ultrasound, and I'll talk about those last two in a bit more detail in a moment. So when we are trialling a new idea in terms of maceration, we usually go to the lab and we try it at a small scale first of all before we come out to industry and tap on somebody's shoulder and say, can we have half a tonne of grapes to play with, please? So we get things right in the lab and we get a sense of whether they're going to work and make a difference that is appealing. So we harvest grapes, crush and destem them by hand, inoculate them with yeast, and then we have been using this, what we call the French press method, developed by Dr. Dambergs. It's a really effective, tidy, food-safe way of simulating a submerged cap fermentation. It's been great. We press the wines off, and we can replicate that, which means we can get statistically valid results out of the trials. We press them off, settle them out. We don't do malo on these smaller scale trials because it confuses the results somewhat, and then we bottle them off and bottle age them. After that we analyse the wines. So for the red wines that we develop in the lab and analyse, we run basic wine analyses. We've also really strongly lent on the phenolics analysis by UV-vis spectrophotometry, the modified Summers method, which gives us 9 different ways of understanding the phenolic profile of the wine. And then for additional analyses, we really lean quite heavily on our collaborators and use contract analytical services. So, for example, we have had some really valuable outcomes from working with the AWRI to look at tannin composition, polysaccharides, also with Metabolomics Australia to look at volatile aroma compounds. And when we're feeling extremely rich, we indulge ourselves in formal sensory analysis, again really beautifully supported by the Australian Wine Research Institute's team there. So this is a photograph of a trial from 2011, I guess one of the first ones that I was involved with, where we were looking at a range of different maceration processes. And I like this picture because you can see quite clearly here at 6 months bottle age we've got very discernible differences in the visual the visual appeal, I guess, of the wines. You can see the names of the treatments that we've applied around here arrayed at the base of the wines. At that time we were particularly interested in time on skins because we were interested in production efficiency. So the 4 glasses on the left, we're talking about 8 days on skins, and the 4 glasses on the right, 12 days on skins. Now 3 of those treatments, high sulfite, enzyme and dry ice maceration, really didn't give us much of an interesting result at that time. So at that point we sort of said, they're probably not very useful, and we turned away from them. But we will come back to those in a moment because we had another look at them. So just focusing in on the remaining 5, I'm just going to show you this one table which is at 30 months bottle age, 3 different phenolic measures here. And what's important here, I guess, is just to look at the control at the top here and to see which of those treatments, after a decent amount of bottle age, showing us some kind of substantial or significant difference in terms of these phenolic measures. So what we can see here is all of the anthocyanin, no difference there for anthocyanin. no statistically significant difference, which is what we would expect in a wine of this age. Really, all of that anthocyanin should have been bound by that time. So the non-bleachable pigment column here, you can see that the microwave treatment, the cold soak treatment, and the extended maceration treatment were all showing a significantly higher level of non-bleachable pigment than the control at this point. Now that's very promising for Pinot Noir, obviously, because we do not want the wine to collapse as it ages in terms of its colour profile. One of the interesting results in this study was that we really hadn't seen a significant difference from cold soaking at 6 months bottle age, and so we looked at that and said, oh, well, you know, in terms of phenolics, cold soaking doesn't look to be that useful. But it would appear, and I won't go into this in detail, but it would appear that we may have had extraction of some particular tannin species or even other compounds that were aiding the formation of non-bleachable pigment from cold soaking that we couldn't really see the effect from at 6 months bottle age. Coming over to the tannin column, again you can see that there were 3 treatments here that showed a significantly higher tannin concentration at 30 months, and this is really important for Pinot Noir because again it's one of the things that we sometimes struggle with just to get a decent tannin profile. So on the basis of that trial and some other, quite other substantial block of work, Wine Australia funded us to look at what we call CPR, controlled phenolic release using microwave on a semi-commercial scale. So on the left-hand side here you can see Dr. Kai Knerzer at the CSIRO with a very gigantic and fairly ungainly looking microwave unit that we have worked with over the last 2 years. It's a conveyor system, so the material goes in this end. These are magnetrons, so there's banks of 3 along each row and there are 5 of them, so it's 15 magnetrons, and then the material comes out the other end. And you can see here Dan Buckle and Keithy, whose first name I don't know, and we're just having a look at the juice as it's come back from being microwaved on the right-hand side compared with the control. So that year was 2014. We ran an industry trial comparing those 2 methods, which was really valuable. The following year we did the same trial, but we pressed the microwaved material off at day 3 or 4, and we kept the control on skins until day 8 or 9. And the graph that you're looking at the moment is called a principal analysis scores plot. And what it does is it takes a large number of data points and compresses them down to 2 dimensions. And so what we can see, I guess the take-home message here is that even when we pressed the microwave wines off at day 3 or 4, you can see that all of those microwave wines are sitting on the right-hand side of the control wines. And in this graph what that means is that the microwave wines had a higher concentration of phenolics. So we got greater extraction even though we had less skin contact time. One of the things that people very reasonably and understandably ask is how do they taste? How do these microwave wines taste? Thus far we have run informal tastings. We really like to get out to industry and let them taste what we're doing. The blind tastings that we've run, the microwave wines travel very well. When people know what they're tasting, they tend to be able to perceive characters in those wines that they often name as stewed or sort of cooked aromas. So, and as I say, the blind tastings work very well because when people can't tell which one's which, usually the wines made by CPR come out on top because of largely because of the tannin profile. You can see on the right there some comments from winemakers about the on-skins treatment and then also about the early press microwave treatment. So those wines will go to formal sensory appraisal within the next 6 months, and we'll be able to say something a little bit more definite about their sensory appeal or otherwise. I said at the beginning of the talk that really we are very cognizant of industry's need for practicality and to be able to justify these kinds of technology choices when we're looking at novel maceration. So for the microwave maceration, we have investigated the business case. I'm going to go fairly quickly through these slides. You're welcome to come back to them, obviously. Mr. Paul Smart put together a business case for these, and he looked at two different methods, winery, new greenfield development, and then the addition of equipment to an established winery. The major saving for the microwave maceration was the labour saving from not having to pump over from that early press off. But there's also, of course, a cost to invest in that machinery or that technology, and there's a saving in terms of decreasing the vintage length, or optimising your capital equipment. He calculated return on investment for the assumed value or cost of a microwave unit, and as you can see there, it's quite it pays itself back quite quickly. So potentially worth considering additional work on that. I'm just going to talk now a little bit more about some of the other maceration treatments that we've looked at. So you'll recall from the early part of this talk that when we examined enzyme in 2011, we really didn't see much of a kick from enzyme in terms of phenolics in wines. So we came back to that partly because we were thinking about cold soak and what the mechanism might be behind some of the results that we'd seen out of cold soak. And we were wondering whether the enzymes that exist within a grape to help with the ripening process, so these are pectin methylesterases and polygalacturinases, whether some of those enzymes might be the reason that cold soaking gives us some of the effects it does, and whether we could simulate that and speed it up by using enzymes that were available commercially. The schematic on the right here, I don't know whether we really need to go into it, but you can see there that's the external surface of the supposed grape, and the material we're interested in mostly is pectin, these pink little dotty lines, because the pectin really binds together all those cellulose microfibrils that really maintains the structural integrity of the skin. So we investigated using enzyme, using cold soak, using no enzyme and no cold soak, and using both enzyme and cold soak. And the very short version of this complicated plot is that we really did not see, at this 8 months bottle age, we really didn't see an effect from either enzyme or from cold soaking. Now this is reasonably consistent with some of the other research that's been published on enzyme and cold soaking, and so I guess I wouldn't say that we could make a conclusion about this, but it's probably worth our while having a bit more of a look at what's going on with enzymes, what's going on with cold soaking, so that we can really see whether that is actually a useful and efficient and effective mechanism or winemaking tool, and give some valuable, or hopefully valuable, insights for industry there. Another part of the early trial that I talked about, the 2011 trial, was that we looked at what we called dry ice maceration at that point. And we certainly were interested in that, but we didn't see a result from that. In 2015, we conducted a second fairly substantial maceration trial with Dr. Kerslake, Dr. Smith, Binden, Dambergs and Close were all involved with that trial. We were interested to scale up to 10 kilo ferments replicated so that we could take them out to industry tastings. We were also interested to investigate some of these novel maceration treatments that we really hadn't looked at closely enough. So during this trial we ran 3 replicates at 10 kilos for each of these treatments. There was a control, a cold soak for 4 days pre-inoculation, extended maceration for 4 days post-inoculation. Now that's not a large extended maceration necessarily by industry standards, but it's enough, we know, to give us a fairly strong result. You can see the photo there, even with the early wine. We used microwave and left the skins in, and we also used microwave with the early pressing. And then we looked also at inline CO2, which I'll explain in a moment, and ultrasound. So they were 2 fairly novel treatments that we were trialling there. So inline CO2 was to simulate a piece of technology we're aware of called the Boreal, which is an air liquide piece of technology. It goes into the must pump line, and I do apologise, this is possibly in Spanish, I stole it from the internet. But it goes into the must pump line, and the intention here is to use CO2 to drop the temperature of the must very quickly when refrigeration is a pinch point in a winery. So it's just a different way of getting it cold. But we were particularly interested in the idea that CO2 could potentially be spot freezing parts of the grape material, and we knew from the freeze-thaw maceration trials that freeze-thaw was actually effective for busting out some of the phenolics we're interested in. So here I'm simulating the CO2 being pumped through, a bit of a blue light disco in a bucket there. The other treatment that we tried during the 2015 maceration trial was ultrasound. Now, I won't go on and on about ultrasound, but it is an absolutely fascinating process. But the basic idea is that we create cavitation inside cells, and this causes cell rupture, and then the idea is to enhance the mass transfer. And in fact, in this schematic, mass transfer is going directly through the skin, but we believe in grape material it actually penetrates into the pulp as opposed to coming out this other way. So you can see top right-hand photograph that one of those buckets has been ultrasounded with my magical ultrasound wand there, and the other bucket's sitting there waiting. Actually, I don't think I treated that one. That was a control. So I'll just go back here. The results we got from the 2015 trial was sufficiently interesting that we were tentatively encouraged to trial it in 2016 at the CSIRO. So this is a trial on Shiraz coming from Shadowfax, which I'm very grateful to them for their allowing me to use their crusher distemper and not being too alarmed at me shoulder lifting 300 kilos of grapes into it. We transported that material after it had been crushed and distempered back to the CSIRO, and either we put one treatment through the inline ultrasound, tunnel ultrasound, and the other one through a batch, more of a batch bucket ultrasound, and sampled, bottled, analysed, and we're currently aging those wines, so they will get, we'll look at those properly at 12 months bottle age. Coming back to the 2015 trial, when we examined those wines at 6 months and 12 months, we really saw, based on these PCA scores plots, that we are getting differentiation, obvious differentiation. So having a look at the green circle there, that's from extended maceration. Down here in the bottom left-hand corner of this plot, the CPR with early pressing. We pretty much know that gives us a pretty big result for phenolics. And over here, the CPR on skins, that's a very strongly extracted wine. But what was perhaps more interesting to us from the 2015 trial was that we were starting to see a bit of differentiation with that inline CO2 and also with the ultrasound. So they're not pulling out very strongly, but we have done some additional analyses on these, in particular with support from Dr. Smith and Bindon. And we are seeing some tannin composition differences for the ultrasound treatment, and we put some of those wines forward at the Australian Wine Industry Technical Conference, and there was some informal sensory feedback that there were mouthfeel and aroma differences from both the ultrasound and the inline CO2 simulation. So I guess that what we have learned from our maceration work, I'm just going to wrap that part of it up and I'll talk about some new projects we have coming up. But what we have learned very strongly from our novel maceration work is that the thermal maceration process that we've been working on with microwave is really very, very effective, but also quite flexible in that we can get a strongly extracted wine if we leave the skins in, which is a good blending component for years particularly where we're not necessarily having good phenolic character in wines. But also we can potentially look at some really significant production efficiencies and a little bit more control over phenolic profile with the early pressing off. So thermal maceration by microwave, or even perhaps thermal maceration in general, is most definitely something that we feel is worth pursuing and considering further. We're also very interested, of course, in the ultrasound and the inline CO2 treatments, because even though the phenolic result is not quite so spectacular, we're definitely getting a phenolic result, and some of the early sensory, informal sensory feedback we're getting is that we may be in fact creating a wine that has quite a nice mouthfeel with those two treatments. I'm just very briefly going to touch on some new projects that we have coming up, and then I'll be really happy to take any questions or comments from you people listening. So we have a strong focus on maceration, on red wine maceration, but also our group's quite strong on sparkling wine. So one of the projects we have just initiating now is led by Dr. Fiona Kerslake, building and measuring the quality of fine Australian sparkling wines through identification of impact compounds responsible for autolytic character and novel winemaking technologies to hasten autolysis. So this project will run for 4 years. It is actually a project in 4 parts. One of the projects is looking at tasting and analysing wines that Dr. Kerslake and others have had in cellar since 2010 based on some viticultural trials, so that's going to be very interesting. Another project is using novel technologies to hasten the lysis of yeast so that we can potentially impact the amount of time that we need to have those wines on lees before they're ready for release. A third project is looking at inline sensing of phenolic pickup in the pressing of juice for sparkling wine production, and we're also looking at discrimination of the provenance of juice for sparkling wine. Another project we just have kicking off is we're part of, Dr. Kerslake is part of a consortium looking at Australia's wine future and adapting to short-term climate variability and long-term climate change. It's led by the Antarctic and Climate Ecosystem CRC, which is based here in Hobart with many partners and should be quite an interesting project. And a project which I will be leading over the next 4 years is in short called Pinot Noir Provenance. The long title is Australian Benchmarking to Support the Growing Making Perception of Quality and Marketing to Add Value to the Pinot Noir Supply Chain. It's funded by Wine Australia. There are multiple institutions, there are industry partners, and we have international partners as well. And really the intention of this project is that based on high quality analysis and big data integration, the project will deliver practical guidance for stakeholders all the way along the value chain. So from investors who are looking at planting, people who are growing Pinot Noir, winemakers and also marketers. So they will be projects to watch over the coming years. But for now, I would like to wrap up by making a most sincere acknowledgement of our funding. Oh, dear me, I do apologise, I've left the Wine Industry Tech Committee in there. The funding body, Wine Australia, Australian Wine Research Institute, a very important collaborator for us, the CSIRO at AWRI, and then really important in-kind and intellectual support from our colleagues in the winemaking industry and grape growers, and also some of our colleagues who contribute to the research. So that's me done. Now, I've had a lesson on how to deal with questions and I'm ready for them at any point now.
Thanks, Anna. I'll just reiterate that to ask a question, please type into the question section in your control panel in the right-hand corner of your screen. And for those with access to a microphone, there is also the option of speaking directly with our presenter by clicking the Raise Hand button. This is located in the top left section of your control panel. Now, we do have a couple of questions. Firstly, Alex has got his hand raised. Alex, I've unmuted your microphone. Do you have a question?
Sorry.
Yes, go ahead. Alex?
Look, I do have a question. There's been a lot of work regarding, obviously, extraction of colour and through the different methods. I suppose for me, the next step is how do we look at stabilising, I suppose, binding these colours up long term? And is that something that is planned, I suppose, work on for the future?
Yeah, absolutely. Hello, Alex. Who are you and where are you from? I don't recognise your voice.
So, Alex Trescothick. I'm based up in the Barossa Valley at the moment with Pinot Ricard at Richmond Grove.
Well, hello. It's lovely to meet you. Yes, I think really one of the real challenges for this work, and something that we thoroughly enjoy looking at is it's all very well to get a kick after 6 months, or when you've just pressed the wines off to see a great big chunky sort of anthocyanin result, but really unless there is a decent relationship between the amount of tannin you've managed to extract and the species of tannin that you've managed to extract, and also the anthocyanin concentration, we're really not seeing decent stabilisation of colour over time. So it is most definitely not a simple equation. For example, one of the pieces of work that Dr. Angela Sparrow has completed, which has been quite important, she's been looking at the different role that what we think of as seed tannin and what we think of as skin tannin play in longer term colour stabilisation for Pinot Noir, and the very early sense is that the seed tannin is not only potentially quite astringent and bitter, but also it may not be as effective in achieving that long-term colour stabilisation. So I guess the very early thought is that if we can optimise or maximise skin tannin extraction in Pinot Noir and manage potentially back off the seed tannin extraction, we may achieve better colour stabilisation. Some other work that we're involved with, but really Dr. Bindon at the AWRI is leading this work, and Dr. Paul Smith, is looking at things like the polysaccharide composition of the wines, because again those polysaccharides potentially look like they're quite important with colour stabilisation. So polysaccharides are coming from two different sources within Pinot Noir. We have grape-derived polysaccharides, which tend to be a bit shorter, and then we have also yeast-derived polysaccharides, which are coming out of the manoprotein portion of yeast, which is manoprotein is in the external cell wall of the yeast. So those two components are looking like being quite important with colour stabilisation, but also in mouthfeel modulation in red wine. So yes, I think, Alex, it's a really, we most certainly don't know the answer to those questions yet, but it's a really important point, not just for winemakers, but also very much for researchers, to understand that the amount of time we give wine to age and settle down, and the way that we look at it as it ages is really important, not just the early results we get from novel maceration processes. So, and you're very welcome to ask me a follow-on question if I haven't addressed what it is that you wanted to talk about.
Oh, no, that was very good. Thank you.
Cool. Thank you.
Thank you, Alex.
And I'm not seeing a button that says questions on my screen.
It's about to come through now.
Oh, good on you. Excellent.
Can you see the question?
Oh, here we go. Yes, I have a question. Thank you, Sarah Burville. It's nice to meet you, even though I can't hear you. Do you see an increase in VA production with microwaving? That is a very interesting question. I'm just thinking back. So, what have we looked at? We did a very interesting trial. Now, this was an interesting trial. So, I'm going to digress slightly, but you'll have to bear with me because it's interesting and also relevant to your question. So when we were doing microwave maceration and early press-off, we noticed that we were making wines that were extremely pretty. They were almost confected, very, very sweet, lolly, a bit sort of red fruit, almost like the style of wines you expect out of thermovinification generally. So we were quite interested in this scientifically, but also interested from an industry perspective, because that style of wine only has a sort of a small market segment, really. Now, what we did then was we ran a trial where we adjusted both the presence and absence of pomace in the fermenting wine. So we pressed some of them off and just fermented them as juice, and others we put some of the grape material back in. We also adjusted the the lid. So wines either had a solid lid or they had a perforated plastic on top. What we found was that there was a VA effect. So actually it was a very complex effect. So it wasn't just VA, there was a really diverse aroma impact from the presence or absence of pomace and the presence or absence of a lid. So the short version of this, I guess, is that the way that we ventilated the ferment was very important in not just VA but all of the aroma compounds which were contributing to this very pretty style of wine. But also the presence of and absence of pomace had a preferential impact on certain aroma compounds. So the pomace appeared to be either contributing or scrubbing certain aroma compounds from the wines. So we don't see obvious unpleasant VA problems with any of the microwave treatments that we use, so that's not an issue, but we definitely see some differences in VA and a lot of aroma compounds depending on how we handle those ferments. And again, Sarah, please feel very free to type in an additional follow-on question if there's anything that I've not managed to cover. I'll come back to it after any other questions. Now, and I can't see any more questions. Oh, wait a minute, I have got another question. So, oh, hello, Neil. Neil Scrimmager has asked a question. It's nice to hear from you. Anna, can you elaborate on the assumptions made in the payback period calculations in regard to viable production savings, lower operational costs from inclusion of the CPR technology? Yes, I'm just going to see if I can find that. Now, can you all, I hope you can, Anne, can you tell me, can you see me going backwards through my slides? Let's assume that you can.
Yes, I can.
Great. I'm just putting the assumptions up again. So this is work that was undertaken by Mr. Paul Smart, who I think is probably at the tail end of finishing his Masters in Wine Business. And the assumptions that he made here were that the wines would be pressed off at day 3. So he was looking at CPR as in that early pressing. And you can see here that what he found was that when we build a winery around that technology, and he was assuming there that all of the reds would go through that process, which is probably, they probably wouldn't. But the extreme case was 62% less fermenters required due to decreased fermentation volume. And this is not rocket science. It's certainly something that we see in Europe where thermovinification is used. So in Germany, we see really very rapid throughput because you don't have that time and that volume taken up with wine fermenting on skins. The point that he makes here in the Greenfield and also in the addition of CPR to an existing winery was just this labour saving. Paul has quite a good record as a winemaker, and the amount of time and energy that goes into pumping over red wines is a massive labour impost for wineries, and so that was a very important saving in his set of assumptions. The full business case, I don't think it's up yet, but it will be in the final report for this project, and that final report will be due in February. Actually, we may get an extension on that. So certainly there's a substantial document, and if anyone's interested in that, I'd be very happy to send you the current draft, which is actually in very good shape. So please let me know. I'd be delighted to get some feedback on it. Now, any other questions? Let's see.
At the moment, there's no other questions sent through. If anyone has a question, if you could send it through now. Otherwise, we'll finalise the webinar.
I feel like I should be perhaps singing and dancing in the interim, but that might be distracting.
Well, maybe while we're waiting, you could perhaps do a little sum up, and we'll see if anything else has come through.
Sure. I'll just I'm just adjusting my screen so I can see where I am. What do I have to say? I'm not really sure that I have much more to say. I guess that what I concluded with, I would just reiterate that I do think that there is most definitely potential for us to investigate thermal technologies within Australia. I'm very aware that some of the challenges with the use of thermovinification are around that production of quite a pretty strawberry aroma type wine, which is only, it's only appealing to a certain amount of people. But some of the work that we've done in aroma moderation and some of the conversations I've had, particularly with Professor Uli Fischer from southern Germany, would suggest that we could probably be very clever in the way that, or we could learn a lot more about the way that we manage those ferments to moderate those or manage those pretty aroma qualities. There's a possibility that those aroma qualities are coming from a heat-based cleavage of aroma precursors. So there's a very simple cleaving of a sugar that happens when you heat grape material, which releases precursors that yeast can then process into those pretty aroma compounds. But some of the work that we've done with covering and pomace readdition would suggest that we can mediate or moderate those pretty aroma compounds. So yes, that's definitely something that would be good to follow up on.
Okay, well, no more questions have come through, so I think we might finalise the webinar. So thank you, Anna. I'm about to take back control.
Great. So maybe I can just say thank you very much to those of you who have listened in, and anyone who listens to this on the internet later on. I'm very happy for you to send through questions or initiate conversation with me. Obviously we're very passionate about this research at the Tasmanian Institute of Agriculture, and we'll have a coffee or a glass of wine with anyone who wants to have a good old chat about it.
Okay, thank you, Anna, for presenting today, and thank you also to everyone who attended and took part in today's session. For attendees, you'll receive a follow-up email with a link to this recording and a link to the survey. The next AWRI webinar is the 6th of October, when Andrew Hall from Charles Sturt University will present remote sensing applications in viticulture, recent advances and new opportunities. If you would like to register for this webinar, please visit the AWRI website, and that's all we have for today. So thank you again for attending, and I look forward to seeing you again at the next AWRI webinar.